Yes, so thank you very much for giving me the opportunity to present our latest results here. You all know and heard yesterday, we have all the canonical bases in our genome, but there's a second layer of information, and these are these epigenetic bases like 5-methylcytidine, hydroxymethylcytidine, homocytidine, carboxycytidine. And you can think about it like every cell gets every information in terms of like a big book, and these epigenetic marks are kind of like posted which parts of the or chapters of the book a particular cell needs at a given time. And with this marks, cell, for instance, regulates which genes needs to be expressed, silently, composed by elements. And these marks also change during our life. And if we become older or cells get disease, so there's a particular interest in identification of these marks at a given time of a different state of the cell. So we're talking about epigenetic clocks that determine our age or if we get a disease and so on. And who are the players that install or regulate, or some of the players install and regulate these marks? For instance, they 5-methylcytidine are installed by different methyltransferases, and then we can have passive demethylation effects, for instance, by during cell division, if there's no de novo methylation happening, so we get a dilution of methylation. On the other hand, you can have active demethylation by PET enzymes, you heard about it from yesterday, that use iron and alpha ketoglutarate to oxidize 5-methylcytidine to hydroxymethylcytidine, homocytidine, carboxycytidine, and these can also then be removed by the basic cyclic repair enzyme DNA glycosylated. So I hope you understand that there's a need to learn about these processes and also about the spatial temporal localization of these epigenetic marks. And by now we have there are a number of sequencing methods that have been developed, largely also relying on the TET enzyme to identify this positional information of these epigenetic marks. Beginning in 20 years ago, like the development of bisulfite sequencing, where you treat your genomic material with bisulfite here on the left-hand side to deaminate cytidine in genomic material that then to uracil that can be later then identified in the sequencing reaction, basically. But methylcytidine is basically not reactive to bisulfite. However, the disadvantage of this method is that it's really, really harsh, and you lose a lot of your genetic material, a lot of information you actually want to have. So in the past 20 years or so, lots of methods have been developed. Other methods have been developed. For instance, if you use TET enzymes to oxidize 5-methylcytidine to carboxycytidine, you can then react them with pyridine borane to the hydrouracil, and here learn basically about the position of this methylcytidine, because this, the hydrouracil will be then later read out as a T in the sequencing reaction. And this is less harsh than treating your material. You also can still treat your material after the oxidation with bisulfite. This is the so-called TAP-SEC. That is the borane sequencing, and here convert the carboxycytidine to uracil again. If you want to learn about the position, for instance, of hydroxymethylcytidine, what you can do, you can treat the genetic material first with a glycosyl transferase that glycosylates hydroxymethylcytidine, and this is then protected from TET oxidation, and then it still reads as a C, and if you compare that, you always have to compare to a reference genome in these cases. You can learn about the position of the hydroxymethylcytidine. Also a method that uses actually 3 enzymes. For one, it's TET, it's glycosyl transferase. For one, to protect hydroxymethylcytidine, oxidize everything that's not oxidized by this TET enzyme. And then treat everything with a deaminase, Apobec, that deaminates methylcytidine and cytidine to uracil. Then you can learn again on these on the position of these epigenetic bases, and this method is called enzymatic methyl sequencing. Recently, the Thomas group developed a method where you directly can read out the position of the carboxycytidine in smart sequencing method, and therefore they developed a really efficient TET enzyme, because a lot of these methods have the problem that TET enzymes, or if you use Tet2 for instance, it's not so stable, it's not so active, but they will develop a really active TET enzyme. And with this, if you oxidize all methylcytidines to carboxycytidine, you can use single molecule retime sequencing with these biosequencers to identify the position of these carboxycytidine directly. And how does this method work? With this biosequencer, you convert your genetic material to this number structure. You have in these little wells, these 0-volt wave guides, there's a little polymerase at the bottom sitting, and which goes round and round and round the number structure. So you have a really high multiplicity, which gives you really high accuracy and precision of your sequencing information. And when the polymerase incorporates a nucleotide, then you get a fluorescent signal. You not only learn which nucleotide is incorporated, you also have a kinetic information. So how long does the polymerase need to incorporate the signal? This is this pulse width, and also how long it needs to incorporate the next nucleoside, this interpulse duration. And with this, well, they found out that if you have carboxycytidine, the polymerase needs longer. So this is, you have a slower kinetic. And if you train the algorithm, you can directly read out the converted 5-methylcytidine via this algorithm. So this is works already great. So I'm a protein biochemist, so I'm really interested in this enzyme. We're working on alpha-ketoglutarate dependent peroxygenases. So I thought, well, maybe let's try to find some other TET enzymes that work very nice. And here I give you an overview how these TET enzymes look. If you learned yesterday, we have 3 different TET enzymes. We have TET 1, 2, and 3. They share a common domain structure. We have also different isoforms. They're located in different tissues and different cells, but they all have a common folio or common domain structure. So the catalytic domain consists of these double-stranded beta helix domain, and that are interspaced by this low complexity. This is a unfolded, unstructured region which controls the function or the activity of the enzyme. And there was a couple of years ago the structure of human Tet2 that used a shorter construct where they replaced this low complexity insert with a glycine serine linker. And if we look at the structure here in gray of this human Tet2 and then compare with the AlphaFold model of Tet3, you see that the active site is highly conserved. So the enzyme Thomas group developed, or the Tet3 variant was based on the structure. It was a mouse enzyme, and here you see the conservation of these Tet3 enzymes also mapped onto the surface of the structure. So we set out now and cloned or designed different variants from different species of these Tet3 enzymes. We can express them in E. coli. So we use the one from Xenopus, the Homo sapiens one, so they share about 80% sequence identity. And you see we can nicely express them and purify them. And then we set out to test the activity. And the method that's also pioneered by Thomas group is using mass spec to quantify non-natural or non-canonical nucleosides. And in order to do that, you need isotopologues that have a different mass but the same physical properties in order to be able to use mass spec to quantify abundant nucleosides. Basically, you use a reference curve with these isotopologues. They enter the mass spec at the same time but have a different mass. And since you know from your calibration curve how much of your isotopolog was in there, you can quantify all the other nucleosides. And when we react genomic material, it can be synthetic DNA, but also genomic DNA, of course, because it's an interesting bit. We digest it to the single nucleoside, inject it in the mass spec, and then you can follow the conversion, for instance, of the 5-methylcytidine to carbonyl-cytidine. So we use that to compare or test the activity of these different Tet3 variants. And here you can see we just plot the quantity of the modified cytidine. You see, so this is on a synthetic DNA oligo with a single 5-methylcytidine, and then you see that we get a nice conversion. The human variant is a bit more lazy than the other ones, and we tested the next on genomic DNA, and we get really, really nice conversion rates of all our Tet variants. We then were asked by the reviewer that we should compare it to commercially available Tet enzymes. So we got the Tet2 from New England Biolabs that sells it in its enzymatic methyl sequencing kit. But of course, you know what's the buffer, what's the concentration. So first we set out to determine the concentration. So roughly using this back and put it on a page there, roughly we can say, well, the Tet2 in the NEB cells is a concentration of 25 micrograms per microliter, and apparently it's also kind of a shortened version of some sort. So usually we use around 10 microgram Tet of our Tet enzyme to treat 1 microgram of genomic DNA. NEB uses 4 microliter. This is then roughly 100 microgram for up to 200 nanogram genomic DNA, meaning there is about a factor of 50. We first set out and say, well, let's take the Tet and use it under our reaction condition, also in their buffer they sell, which we don't know the composition, and in our reaction buffer. And if you see here, like if we use just 10 microgram of the enzyme, 1 microgram of genomic DNA, we just only get 2.6% conversion rate. If we use more Tet2, we get a higher conversion rate, but also it seems to work better actually in our reaction buffer. Then to be fair, we tested it on the conditions they use, basically 4 microliter with 200 nanogram of DNA. In our buffer, we get here also then almost 90% conversion rate. So also what's also of great interest, because if you use Tet enzymes, you throw in iron and alpha-ketoglutarate, do we get any oxidative side products? For instance, guanine is the base with the lowest redox potential. And what you see here is that if we have an oxygen form, then this can code as a thymine then in the end, or you can get basically a mutation. And here we see that we get an increase by a factor of 2.5. And we also looked at the formation of 5-hydroxy methyluridine. And here we see also an increase. This could be maybe enzyme driven, that maybe once in a while the Tet3 also oxidizes the thymine, but this does not actually change the coding potential. To sum up, so to know where and when particular epigenetic modifications are present is really important to understand aging differentiating disease states. We developed a series of high-performance Tet enzymes based on Tet3, and they all show high activity. And Tet2 shows in our reacting conditions significantly lower oxidation activity, even when we use much higher enzyme concentration. We form a little amount of 8-oxygen and 5-hydroxyl uracil as side products. And Thomas recently founded a company, so these enzymes are also commercially available. Yes, thank you. And I also have to thank my group. This is a picture of former and current group members, and also, it was a great collaboration between our groups. Thank you for your attention.